Our second SoftChalk lesson in Unit 1, Lecture 2 deals with sizes, shapes, arrangements, and forms of bacteria. Now, these are the same objectives you're also going to have in Lab 1 in your laboratory portion of the course. We need to know those throughout the course in both lab and lecture. So let's start out again with our fundamental statements or bullet points. There are three basic shapes of bacteria. Those three shapes are coccus, bacillus, and spiral. A coccus is a spherical or oval bacterium, and based on planes of division, the cocci can appear in several distinct arrangements, namely Diplococcus, Streptococcus, Tetrad, Sarcina, and Staphylococcus. A bacillus is a cylindrical or hot dog-shaped bacterium that can appear as a single bacillus, a streptobacillus, or a coccobacillus. And then a spiral shape is a curved or spiral-shaped bacterium that can appear in one of three forms, either a vibrio, a sporillum, or a spirochete. And finally, remember the metric unit we use to measure bacteria is the micrometer. A micrometer is 1/1,000,000th or 10 to the minus 6 meters. And looking at our detailed learning objectives for this lesson, when you're finished with this, you should be able to list the three basic shapes of bacteria, list and describe 5 different arrangements of cocci, define and give the abbreviation for the metric unit of length termed micrometer, and state the average size of a coccus-shaped bacterium and a bacillus-shaped bacterium. List and describe 3 different arrangements of bacilli, and finally list and describe 3 different spiral forms of bacteria. So those are our bullet points and our detailed learning objectives for this SoftChalk lesson. So keep in mind that as we've already learned, bacteria are prokaryotic, and keep in mind we're talking about the domain bacteria here, which we'll be talking about through the rest of Unit 1. So as a general rule, bacteria are single-celled microscopic organisms. There are a few exceptions. There's a very few bacteria that are just visible to the naked eye. Some of these are listed there, but these can get pretty good size in bacteria where we can just barely see them. But the bacteria we'll be dealing with will all be single-celled microscopic organisms. And of course, as we learned in our prokaryotic, eukaryotic SoftChalk lesson, generally bacteria are much smaller than eukaryotic cells. And this is a picture we showed you earlier of a human epithelial cell from the mouth in Figure 1. So we see how big the eukaryotic cell with its nucleus is compared to the prokaryotic bacteria that are microbiota living on that eukaryotic cell. Bacteria are very complex despite their small size. Even though they're single-celled organisms, they communicate with one another through a process called quorum sensing that we'll learn about in Unit 2. And in this way, they can also function as multicellular populations rather than individual bacteria. So we'll learn more about that later on. But keep in mind, each bacterium is a complete living organism, not just a single cell. In terms of size comparisons, we showed you these 2 videos back when we were introducing the bacteria in our first SoftChalk lesson. And you might want to review that again to get an idea of just how small the bacteria are compared to other things. So there's 2 nice videos there you can look at to compare sizes. So bacterial shape is determined primarily by some unique proteins that form a spiral band, kind of a simple cytoskeleton around the interior of the cell, just under the cytoplasmic membrane. And the way these proteins are recruited and the way they're laid out determines the different bacterial shapes as they grow. But we can divide the bacteria into 3 basic shapes. So again, if we ask for a shape, it's one of these 3 words, either a coccus, which is a spherical or oval bacterium. The plural of coccus is cocci. A bacillus, a cylindrical or hot dog shaped bacterium. The plural of bacillus is bacilli. And finally, spiral, a helical or corkscrew shaped organism. And let's start out with the coccus shape. So as we said, cocci are spherical or oval bacteria, but often when they divide, they remain attached in one of several distinct arrangements unique to that organism. And this is based on their planes of division. So these are the different arrangements of cocci that you'll need to know and the terms we use to describe them. We have Diplococcus, Streptococcus, Tetrad, Sarcina, and Staphylococcus. And these arrangements are showing in the drawing here in figure 2. But as we'll be stressing in lab, keep in mind that when you make slides of microorganisms and view them under the microscope, some of the arrangements break apart and sometimes the arrangements clump together. So you have to look fairly carefully to determine the correct arrangement of cocci. So let's look at each of these different arrangements now. If the cocci divide in one plane, like cutting a ball in half, that's going to produce either a Diplococcus or a Streptococcus arrangement, depending on how many bacteria remain attached. So Diplococcus, diplo means two, these are cocci arranged in pairs. In figure 3, we see a photomicrograph of Neisseria gonorrhoeae, which is a Diplococcus. So you see pairs of cocci there. Now you also see single ones because when you make the slide, some of them do break apart. But the predominant arrangement there is Diplococcus. And figure 4 shows a scanning electron micrograph of another Diplococcus despite its genus name, Streptococcus pneumoniae. And here's a transmission electron micrograph of Streptococcus pneumoniae as well, showing the cell dividing and remaining attached in pairs. A Streptococcus arrangement are cocci arranged in chains. Figure 6 shows a photomicrograph of Streptococcus pyogenes, and you see chains of varying lengths that are produced here. So these tend to remain in chains longer than 2, but keep in mind some of them do break apart, so we see some pairs and some singles as well. Figure 7 is a scanning electron micrograph of Streptococcus pyogenes. That's the bacterium that causes strep throat. So again, we see it's a chain of cocci. So that's division in one plane, Diplococcus or Streptococcus, depending on how long or how many bacteria remain attached. Now, if the bacterium divides in 2 planes, that gives us a tetrad arrangement. So a tetrad arrangement is cocci arranged in squares of 4. And in figure 8, we see that very nicely here in this indirect stain of Micrococcus luteus. So again, we see lots of little squares of 4 bacteria here. Now again, some of them break apart, and sometimes when you make the slide, the bacteria don't separate, they remain stuck to one another in big clumps. All slides show clumps, as we'll stress in lab, but the predominant arrangement there you see is a tetrad, a square of 4. And even in the clumps here, you see the clump is made of squares of 4. And this is a scanning electron micrograph of several tetrads as well, where you can see 4 bacteria arranged in some of the cases here. If the coccus divides in 3 planes, that gives us a Sarcina arrangement. So a Sarcina arrangement would be cocci arranged in cubes of 8, 4 stacked on top of 4. Figure 10 shows you a photomicrograph of a Sarcina. Now it's very hard using the microscopes we have to tell a Sarcina arrangement from a tetrad arrangement, because you're looking down on top of a square of 4 or a cube of 8, which looks pretty much the same. So about the only way you can really tell a Sarcina from a tetrad is to be able to actually count the planes of division. And with the oil immersion lenses we have, that isn't really possible. You need a very expensive oil immersion lens with great resolution to do that. So basically, anytime you see under the microscope what looks like a square of 4 or could be a cube of 8, you just say it's a tetrad or sarcina. You don't have to tell those are two-part under the microscope, but you have to know the difference between them, that one is a square of 4, tetrad, the other is a cube of 8, sarcina. And then if division is in random planes, that produces a staphylococcus arrangement. Staphyle actually means a bunch of grapes. So these are the ones that produce grape-like clusters under the microscope. So a staphylococcus arrangement are cocci arranged in irregular, often grape-like clusters like we see in figure 11 and 12. So you can see here that there's lots of nice little grape-like arrangements here. Again, there's large clumps where the bacteria didn't separate when you spread the bacteria out on the slide. We see that some of the staphylococcal arrangements have broken apart, so you often see pairs and short chains and singles, but the predominant arrangement we see here are small clusters, often grape-like. And the key here is that a staphylococcus arrangement is not going to be any more than maybe 4 or 5 bacteria across. Anything bigger than that, just a clump of bacteria. And again, all slides have clumps of bacteria. That's not an arrangement. So Staphylococcus is a specific cluster. It's one that's only maybe 4 or 5 at the most bacteria across, and often they tend to taper like grapes. And again, here we see several grape-like arrangements of Staphylococcus in figure 12. Under a scanning electron microscope, we see Staphylococcus aureus with typical grape-like clusters. And in figure 14, some methicillin-resistant Staphylococcus aureus, or MRSA, where again we see a lot of nice little grape-like clusters of bacteria. Now, the average size of a coccus-shaped bacterium is about 1/2 to 1 micrometer in diameter. And when we see these magnified 1,000 times with our light microscopes in lab, an average coccus is going to look about the size of a period on a printed page. So they're very small. And there's a little self-check you can do there on arrangements. Our second shape is the bacillus, sometimes called a rod shape. So these are cylindrical, rod-shaped, or hot dog-shaped bacteria. They divide in one plane, producing one of 3 arrangements: bacillus, streptobacillus, or coccobacillus. So most bacilli appear as a single bacillus. And remember, bacteria divide by binary fission. One cell splits in 2. So you have to be careful in the lab in recognizing a bacillus, because if it's an actively dividing culture that you're staining, a lot of the bacilli will be undergoing binary fission, and a small bacillus undergoing binary fission can look very similar to a diplococcus. So anytime you see what looks like a diplococcus or single coccus, you have to carefully look around for these little hot dog shape to make sure that that is in fact a bacillus undergoing binary fission, and you're not mistaking it for a diplococcus. Some bacilli are arranged in chains. Strepto means chains, so streptococcus up above would be cocci in chains. Streptobacillus would be bacilli in chains. And a coccobacillus is a very small bacillus that looks very similar to a coccus, but it does divide and lay down cell wall like a typical bacillus. You won't have to recognize any coccobacilli under the microscope, but you should know what a coccobacillus is, that it is a very small bacillus, similar in shape to a coccus. So looking at a single bacillus or single rod, this is a photomicrograph of E. coli in figure 16 that we'll be seeing a lot during the semester. And again, you see these nice little bacillus-shaped bacteria. They look like little hot dogs all over the place. Now you notice some of them are dividing, and so a dividing bacillus can look similar to a coccus if it's just divided, or a diplococcus if it's in the process of dividing. But again, if you look carefully there, you do see lots of little hot dogs here, so you know that is a bacillus and not a diplococcus. This is a scanning electron micrograph of Pseudomonas aeruginosa, another bacillus-shaped bacterium. So again, they look like small corn dogs, kind of. Here's a scanning electron micrograph of E. coli, where you see the nice bacillus shape. And again, you see some on the process of dividing by binary fission. A streptobacillus are bacilli arranged in chains, as we see in figure 19. This is a gram stain of Bacillus megaterium, which is a streptobacillus arrangement. So you see it's a number of bacilli attached to produce long chains. And again, a coccobacillus is very similar to a coccus. This is Acinetobacter, a coccobacillus. So again, you see that they look very similar to a coccus. And again, you won't have to recognize a coccobacillus in the laboratory. But here's what they do look like. Now, an average bacillus has a width of about 0.5 to 1 micrometers and typically a length of about 1 to 4 micrometers. So again, these are just a few millionths of a meter in size. And often a bacillus looks like a tiny dash under the microscope, like we see with our E. coli here. And our final shape is the spiral shape. Now, because these are all single-celled and they don't remain attached, we call these forms rather than arrangements. And the three spiral forms are Vibrio, Spirillum, and Spirochete. We see an illustration of them in figure 22 here, where Vibrio looks like kind of a bent rod or a curved rod. A spirillum, as we'll see, is a thick, rigid spiral, whereas a spirochete is a thin, flexible spiral. So looking at a Vibrio, again, these are curved or comma-shaped bacterium. And here we see in figure 23 a photomicrograph of Vibrio, which looks like a curved rod. And here's a scanning electron micrograph of Vibrio cholera that causes cholera. And again, they look like curved or bent rods. A spirillum, on the other hand, is a thick, rigid spiral. And figure 25 shows you a photomicrograph of spirillum like we'll see in lab. And as you see, these are very thick bacteria. They're easy to measure the width, and they do occur in spirals. And the spirals are quite regular looking. Now, in figure 25, here you can still see the spirals very nicely on the spirillum species. But often when you make the slide, as the slide dehydrates and as you heat fix it, the spirals flatten out, so they tend to look kind of S-shaped, like we see here. So that's when the spirals flatten out. So sometimes you can actually see the spirals in them under a regular light microscope. But oftentimes it'll look more like the bottom bacterium here. It's kind of a nice, thick, wavy organism. But again, they're very thick, and the spirals are very regular and rigid. Whereas a spirochete is a thin, flexible spiral. Under a conventional microscope, a spirochete looks like you took a very fine pointed pencil and drew a little very thin squiggly line. As we see in figure 26, a photomicrograph of the syphilis spirochete, Treponema pallidum. So you see how much thinner that is than the spirillum up here in figure 25. And you see how much more flexible looking it is. The spirals are less regular. It's a much more flexible organism. So those are the three forms of spirals: Vibrio, Spirillum, and Spirochete. Here's a scanning electron micrograph of Leptospira. So again, a very thin spiral-shaped organism. A scanning electron micrograph in figure 28 of Treponema pallidum, the syphilis spirochete. Now, these can get quite large when it gets to spirillum and spirochetes. A Vibrio is more the size of a bacillus. It can be as small as 1 micrometer long, but some of these spirillum and some of the spirochetes can get over 100 micrometers long. So these are quite large as bacteria go. Now, during bacterial division, in the center of a bacterium are a group of proteins called FTS proteins. stands for filamentous temperature sensitive proteins, which you don't have to know. But these are the proteins that interact and form what's called a Z-ring in the cell division plane. So it's these FTS proteins that form the cell division apparatus called the divisome that are involved in bacterial binary fission. So in figure 30 in this illustration, we see a coccus that's already divided its DNA. So we see the nucleoid. And in the center, when it gets ready to divide, is actually a ring of proteins called FTS proteins. And these all work collectively to divide the bacteria in two during binary fission. Figure 29 on the left that we looked at under the pro and eukaryotic cell shows you a bacillus dividing. So we see again that the DNA has already replicated. It's made a copy of its chromosome. One chromosome has been pulled to each half of the cell. And now this is where the divisome is. That's where the FTS proteins are going to form a ring. They're going to synthesize a cell septum. Of cell wall divides bacterium in two. The cytoplasmic membrane will invaginate and pinch in to eventually divide the bacterium in two. So when this process is completed, it'll look like this division plane off here to the left. Here's another bacillus in the streptobacillus getting ready to divide. These are not organelles, by the way, these round structures. These are artifacts that sometimes appear during cell division when preparing it for viewing under an electron microscope. Now, there are some exceptions to those three shapes. So again, our three main shapes are coccus, bacillus, spiral, and those are the shapes we'll be looking at during the semester. But there are some bacteria that are surrounded by a sheath. Others have little stalks they use to attach to surfaces. Some, like the actinomycetes in the soil, produce long filaments similar to a mold, but they are prokaryotic. There's a few bacteria that are actually square-shaped. There's some that are star-shaped. There's some that are spindle-shaped. Some divide longitudinally, forming what are called trichomes. We see that in this transmissional electron micrograph of Liceella filiformis, where notice it's dividing longitudinally rather than transverse like typical bacilla. Here's a filamentous bacterium to the right of that, where you see long filaments that just keep growing longer and longer. And there's even some ultra small bacteria that have been discovered in groundwater. We're not going to be too worried about those, but there's some of these are down to the size of larger viruses, actually. So anyway, those are our three shapes of bacteria, the 5 different arrangements of cocci, the 3 different arrangements of bacilli, and the 3 spiral forms. And again, we have to know those shapes, arrangements, and forms throughout the semester in both lecture and lab, as well as the approximate size of many of these microorganisms. And there's a little self-check you can do there on this section. And a self-quiz at the end covering the whole section.